Hi, my name is H2A.Z and I'm an assistant professor at the University of Toronto in Mississauga. My lab is interested in the epigenetic basis of learning and memory, and today I'm going to provide a speculative interpretation of our recent discovery that the histone variant H2A.Z is a novel regulator of learning and memory in rodents. In the lab, we model memory by using the contextual fear conditioning paradigm, which simply involves taking the mouse, placing it into a novel environment, and exposing it to a mild foot shock, allowing it to create a context and shock association. Using this model, others have shown that neural circuits underlying memory formation change over time. So whereas memories are initially dependent on the hippocampus, over time they're downloaded to the cortex for long-term maintenance. We know that memory formation is highly dependent on changes in transcription, and epigenetic mechanisms are critical regulators of transcriptional activity. Various branches of epigenetics have been investigated in relation to memory, but histone variant exchange is an entire branch of epigenetics that has never been studied in relation to cognitive function. So our goal was to investigate whether histone H2A.Z, a variant of histone H2A, is a novel regulator of memory formation. For our purposes, we decided to split memory up into two main phases. The first is the phase occurring in the hippocampus immediately after learning, and the second was a delayed phase occurring in the cortex. Using this approach, we showed that histone H2A.Z is rapidly exchanged in response to fear conditioning. And specifically, we showed that H2A.Z eviction from the +1 nucleosome was associated with increased expression of memory-promoting genes. What this suggested to us then was that H2A.Z eviction from the +1 nucleosome is very important for regulating stimulus-induced gene activity. So, consistent with this interpretation, we found that depleting histone H2A.Z using a virally mediated knockdown further enhance memory formation and enhance the expression of certain memory-promoting genes, suggesting that removal of H2A.Z is beneficial for memory. So we'll now shift our focus to delayed time points after learning as the memory becomes increasingly dependent on the cortex. So what's interesting is that at the 7-day time point, we now see that H2A.Z is no longer evicted from the +1 nucleosome like it was during the stimulus-induced phase, but it is in fact incorporated into the -1 nucleosome, suggesting that this new modification may be important for the prolonged process of downloading memories to the cortex. So we were really excited about this finding because it provided a long-lasting modification in the brain region that's absolutely critical for maintaining memories over time. So what could H2A.Z be doing at these memory-promoting genes 7 days after learning? Well, our hypothesis that we present in our paper is that H2A.Z at this time point is acting as a marker for genes that need to be reactivated in order for the memory to become stable over time. So ultimately what we think H2A.Z is doing is providing a way for genes to remember their own activation history so they can stabilize memories that allow us to remember our history.